The everyday activity of eating involves more risk than you might think. It is estimated that each year in the United States, 48 million people suffer from foodborne diseases. 128,000 of them are hospitalized and 3,000 die. In the developing world, contaminated food and water kill over 500,000 children a year. In most cases, virulent types of bacteria are to blame. Bacteria are an integral part of a healthy life. There are 200 times as many bacteria in the intestines of a single human as there are human beings who have ever lived. Most of these bacteria help with digestion, making vitamins, shaping the immune system, and keeping us healthy. Nearly all raw food has bacteria in it as well, but the bacteria that produce foodborne illnesses are of a different, more dangerous kind. Bad bacteria. Many of the bacteria that produce foodborne illnesses are present in the intestines of the animals we raised for food. When a food animal containing dangerous bacteria is cut open during processing, bacteria inside can contaminate the meat. Fruits and vegetables can pick up dangerous bacteria if washed or watered with contaminated water. A single bacterium, given the right conditions, divides rapidly enough to produce billions over the course of a day. This means that even only lightly contaminated food can be dangerous. Bacteria can also hide and multiply on dish towels, cutting boards, sinks and knives, and kitchen counters where there is easy transfer to foot or hand. Changes in the way in which farm animals are raised also affect the rate at which dangerous bacteria can spread. In the name of efficiency and economy, fish, cattle, and chickens are raised in giant factory farms which confine large numbers of animals in tight spaces. Cattle, for example, are crowded together under such conditions that if only one animal is contaminated with the virulent bacteria E. coli 0157H7, it will likely spread to others. Tracking the source. Disease investigators like Patrick Griffin are working to find the sources of these outbreaks and prevent them in the future. Griffin of the Centers for Disease Control and Prevention, CDC, in the United States, has worked in the foodborne disease business for 15 years. Periodic E. coli outbreaks turned her attention to the public food safety threat that exists in the restaurant and in the food production system. Food safety is no longer just a question of handling food properly in the domestic kitchen. Now, Griffin says, we are more aware that the responsibility does not rest solely with the cook. We know that contamination often occurs early in the production process, as steps on the way from farm or field or fishing ground to market. Griffin's job is to look for trends in food-related illness through the analysis of outbreaks. Her team tried to identify both the food source of an outbreak and the contaminating bacteria. To link cases together, the scientists used a powerful tool called PulseNet, a national computer network of health laboratories that matches types of bacteria using DNA analysis. PulseNet allowed scientists to associate units in California, say, with one in Texas, type together what might otherwise appear as unrelated cases. Then it's the job of the investigators to determine what went wrong in the food journey to the table. This helps them decide whether to recall a particular food or to change the process by which it's produced. In January 2000, public health officials in the state of Virginia noted an unusual group of patients sick with food poisoning from salmonella. Using PulseNet, the CDC identified 79 patients in the 13 states who were infected with the same type of salmonella bacteria. 15 had been hospitalized, 2 had died. What was the common factor? All had eaten mangoes during the previous November and December. The investigation led to a single large mango farm in Brazil, where it was discovered that mangoes was being washed in contaminated water containing a type of salmonella bacteria. Salmonella contamination is a widespread problem. Salmonella cases involve contaminated chicken, melons, coconut, and cereals were reported in 2018. The mango outbreak had a larger lesson. We no longer eat only food that is in season or that is grown locally. Instead, we demand our strawberries, peaches, mangoes, and lettuce year-round. As a result, we are depending more and more on import. Eating food grown elsewhere in the world means depending on soil, water, and sanitation conditions in those places, and on the way in which the workers farm, harvest, process, and transport to the food. Reducing the risk. There are a number of success stories that provide hope and show us how international food production need not mean increased risk of contamination. Costa Rica has made sanitary production of fruit and vegetables a nationwide priority. Fresh fruit and vegetables are packed carefully in sanitary conditions. Frequent hand washing is compulsory, and proper toilets are provided for workers in the field. Such changes have made Carmela Vazquez, a food scientist from the University of Costa Rica, optimistic about the future. The farmers we've trained, she says, will become models for all our growers. In Sweden, too, progress has been made in reducing the number of foodborne diseases at an early stage. Swedish chicken farmers have eliminated salmonella from their farms by thoroughly cleaning the area where the chickens are kept, by using chicken feed that has been heated to rid it of dangerous bacteria. Consequently, the chickens that Swedes buy are now salmonella-free. These successes suggest that it is indeed feasible for companies and farms to produce safe and sanitary food while still making a profit. battle for biotech, genetic engineering (GE) of crops and animals through the manipulation of DNA is producing a revolution in food production. The potential to improve the quality and nutritional value of food we eat seems unlimited. Such potential benefit notwithstanding, critics fear that genetically engineered products, so-called biotech foods, are being rushed to market before the effects are fully understood. Question: what exactly are biotech foods? Biotech foods are produced from animals and plants that have been genetically altered. Genetic alteration is nothing new. Humans have been altering the genetic traits of plants for thousands of years by keeping seeds from the best crops and planting them the following years, and by breeding varieties to make them taste sweeter, grow bigger, or last longer. In this way, we've transformed the wild tomato from a fruit the size of a small stone to the giant ones we have today. On the other hand, the techniques of genetic engineering are new and different. Conventional breeders always used plants or animals that are related or genetically similar. In doing so, they transferred tens of thousands of genes. In contrast, today's genetic engineers can transfer just a few genes at a time between species that are distantly related or not related at all. There are surprising examples. Rat genes have been inserted into lettuce plants to make a plant produce vitamin C. Moth genes have been inserted into apple trees to add disease resistance. The purpose of conventional and modern techniques is the same: to insert genes from an organism that carries a desired trait into one that does not. Several dozen biotech fruit crops are currently on the market. Among them, varieties of corn, soybeans, and cotton. Most of these crops are engineered to help farmers deal with common farming problems such as weeds, insects, and disease. Are biotech foods safe for humans? As far as we know, so far, problems have been few. In fact, according to a 2016 report from the National Academy of Sciences in the United States, no differences have been found that indicate a higher risk to human health and safety from the GE foods than their non-GE counterpart. Some GE food might even be safer than non-GE food. Corn damaged by insects often contains high levels of fumonisin toxins that grow in the wound of the damaged corn. Lab tests have linked fumonisms with cancer in animals. Studies show that most corn modified for insect resistance has lower levels of fumonisms than conventional corn damaged by insects. However, biotech foods have had problems in the past. One such problem occurred in the mid-1990s when soybeans were modified using genes from a nut. The modified soybeans contained a protein that causes reactions in humans who are allergic to nut. While this protein was discovered before any damage was done, critics fear that another harm proteins created through genetic modification may slip by unnoticed. Moving genes across dramatically different species, such as rat and lettuce, also makes critics nervous. They fear something could go wrong either in the function of the inserted gene or in the function of the host DNA, with the possibility of unexpected health effects. Can biotech food harm the environment? Most scientists agree that main safety issues of GE crops involve not people but the environment. Alison Snow, a plant ecologist at Ohio State University, worries that GE crops are being developed too quickly and released before they have been adequately tested. On the other hand, advocates of GE crops argue that some genetically modified plants may actually be good for the land by offering an environmentally friendly alternative to pesticides, which can pollute water and harm animals. Far fewer pesticides need to be applied to cotton plants that have been genetically modified to produce their own natural pesticides. While applied chemical pesticides kill nearly all the insects in the field, biotech crops with natural pesticides only harm insects that actually try to eat those crops. Can biotech foods have fit the world? 800 million people on this planet are malnourished, says Chanapana Prakash, a native of India and a scientist of Turkish University's Center for Plant Biotechnology Research in the USA. And the number continues to grow. P and many other scientists argue that genetic modification can help address the urgent problems of food shortage and hunger by increasing crop quantities. Crops can be engineered to grow in areas with harsh and dry climate or in soils that are not usually suitable for farming. According to the World Health Organization, an estimated 250 million children in the world suffer from vitamin A deficiency. Between 250,000 and 500,000 go blind every year as a result, with half of those children dying within a year of losing their sight. Golden rice, a biotech variety named for its yellow color, it's thought by some to be a potential solution to suffering and illness, to the suffering and illness caused by vitamin A deficiency. Other experts, however, claim that the biotechnology industry has exaggerated the benefits of golden rice. Golden rice alone won't greatly diminish vitamin A deficiency, say Professor M.N. Marion Nestle of the New York University. Beta-carotene, which is already widely available in fruit and vegetables, isn't converted to vitamin A when people are malnourished. Golden rice does not contain much B-car, and whether it will improve vitamin A levels remains to be seen. What's next? Whether biotech foods will deliver on their promise of eliminating world hunger and improving the lives of all remains to be seen. Their potential is enormous, yet they carry risks. If science proceeds with caution, testing new products thoroughly and using sound judgment, the world may avoid the dangers of genetic modification while enjoying its benefits.